but I'm going to move on to the next topic. I think we only have 2 topics left, electrolytes and osmosis. Okay, so electrolytes. You guys have heard this term before. When you buy like a sports drink or something, you talk about the electrolytes in your sports drink. So it says ions dissolved in solution are called electrolytes because the solution can conduct electricity. Once ionic compounds are dissolved or disassociated is what I'm going to write. I think there's 2 S's there. Disassociated, we talk about the ions, not the compounds. Okay, and this is especially true for blood because our blood contains lots of ionic compounds in it, or lots of ions, such as sodium and chloride and potassium and magnesium and calcium. And so I just want to prove this point to you that when we're talking about electrolytes, sorry if you can hear all that noise. My office is down by all the instruments. Look at this picture I have down at the bottom. I have like 2 beakers of water. And what if we added 2 different substances, sodium chloride and potassium bromide? And then what if we added potassium bromide and sodium bromide and potassium chloride? These are 2 different substances. But once we put them all in solution, they disassociate. So you end up with sodium. I don't know if you can see red very well. You end up with sodium. You end up with potassium. It's all disassociated. You end up with sodium and chloride. And then you also end up with potassium and bromide. So you got these arrangement of ions. These are the only ions present, no matter how much of the stuff you dissolve. And then in the other one, sodium bromide, once it disassociates, you end up with sodium, and you end up with, I keep wanting to say potassium, bromide. And then you also end up with potassium, and you end up with chloride. Now look at the beakers. If you notice, once everything's dissociated in ion form, the solutions are exactly the same. So this is emphasizing the point that as electrolytes, we could dissolve 2 different substances but end up with the same ions in solution, because when we're talking about electrolytes, we're just acknowledging the ions, not the compounds that they came from. Okay, the next thing to know about electrolytes is that there are different there's different strengths to electrolytes. We have strong, weak, and substances that we call non-electrolytes. This is in section 9.8 of your textbook. I encourage you to go read it. This figure, I know it's kind of grainy, is right from your textbook. And so I want to walk you through what is a strong electrolyte. Okay, so sodium chloride is a solid. It's an ionic solid. When we put it in water, it completely dissolves, right? Which is what it says, dissolves in water. I want you to notice this arrow is a one-way arrow. We don't have a reversible arrow, which means this reaction happens one way. Okay, we're not going to get the reverse reaction. We're not going to get these ions coming back together to form sodium chloride. So when you have a one-directional arrow, that means complete ionization, okay? Which is what I want to highlight here. Complete, I probably should use this color. Completely ionized. That is a strong electrolyte. Something that completely ionizes is a strong electrolyte. So you get all ions as your product, okay? All sodium and chloride ions. Okay, a weak electrolyte is a little bit different. Here we have acetic acid. And this is a weak acid. We haven't discussed acids and bases yet. That's chapter 10. But it's a weak acid, and it says dissolves in water, but what do you notice? This is going to reach an equilibrium. We've got a reversible arrow, which means we have the forward reaction into ions, and we're also going to get the reverse reaction where these things come together and remake acetic acid. So at equilibrium, you're going to have some acetic acid, and you're going to have some ions, okay, which is what I'm going to write, some ions, okay, and that happens specifically when you have this double arrow, indicating you have some sort of equilibrium between your reactants and products. That's a weak electrolyte. A non-electrolyte would be like this reaction right here. So we've got glucose. Glucose is not an ionic compound, okay? It will not dissociate in any form. You might have a bunch of molecules of glucose packed together, okay? Think about like a sugar cube. And when you put that sugar cube in some hot water or like hot tea or something, they might dissolve, but they don't dissolve into ions, okay? They just dissolve into individual glucose molecules, okay? So we get no ions, okay? Obviously, ions have charges. You guys know that. We have no charge when glucose dissolves. It does not, a glucose molecule itself does not break up into ions, okay? And then two, double arrow here indicating that you have some sort of equilibrium between our aqueous glucose and our solid glucose. So I had this awesome QR code. I was going to show you guys these three. You know what? I actually think I still want you to scan this and put your answers in so that I could talk about it the next time we meet for lecture. Or just see how well you guys understand what I just said. Yeah, so instead of me walking you through this, pause the video, scan the QR code, do this I want to know this H3PO4, insulin, and HCl, which one is a strong electrolyte, a weak electrolyte, or a non-electrolyte, okay? Yeah, go ahead and do that. All right, hopefully you did. And hopefully, I'm trying to decide if I want to go over the answers right now or not. Yeah, I probably will. Okay. So if you notice on number 1 here, we have a double arrow. We're going to reach an equilibrium, but we do have ions. Oh, I forgot to write this in. This was a negative. Okay, we do have ions on the product side. So some of y'all may be confused that I had no charge on the anion. So we're going to get some ions, okay, which matches this weak electrolyte here. So this will partly ionize. So this is a weak electrolyte. All right, insulin is a solid. It's a biological molecule similar to glucose, and it will dissolve in water, but on the product side, we get no ions. Okay, so this must be a non-electrolyte. And then number 3, we have HCl, a one-directional arrow, and we get all ions. This completely disassociates, completely dissolves for our products. No equilibrium here, so this must be a strong electrolyte. Okay. Notice that a lot of the biological molecules like insulin, glucose, fructose, sucrose, things like that are going to be non-electrolytes. Okay. All right, that's electrolytes. Now, considering we were just talking about ions and charge, kind of connected to that idea is this term equivalence, which we abbreviate capital E lowercase q. So do not confuse this with equilibrium. Equilibrium is usually capital EQ. Equivalence, same letters here, but capital E lowercase q, equivalence. So what is an equivalence? When we have a solution, sometimes we can talk about it in terms of equivalence instead of terms of like molarity or percent. So an equivalent is the number of ions that carry 1 mole of charge. 1 mole of charge. And I'm going to unpack that in just a minute. Okay, so just hang with me, even if you're like, I don't know what a mole of charge is. So why are equivalents important? And then I'm going to unpack what a mole of charge is. Well, our blood contains, or I should say maintains, a certain concentration of charge. Okay, our blood has lots of electrolytes in it, and it's important that the charge balance, like the amount of positive charge from the positive, the cations, and the amount of negative charge from the anions in our blood are maintained. And I actually have a table right here to show you what those concentrations are of positive and negative charge, okay? I'll come back to that in a second. Anybody ever heard of hydration therapy? Okay, so this is another reason why equivalents are important. Let's say that you get really sick and you threw up all night long and your body is depleted, okay, especially for children who have small bodies. What might I then give my son or daughter if that's the case and their body is dehydrated? Okay, a lot of people in third world countries that get diseases from drinking infected water or bad water. They have a lot of diarrhea and their body is in a constant state of dehydration. And hydration therapy is what they can use to balance out the electrolytes in the body. Okay, so you ever heard of Pedialyte? I bet you have. They even have it for adults. If you look on a bottle of Pedialyte, or if you Googled the nutrition label for Pedialyte, it will measure out the concentrations of the ions in equivalents, okay? You'll see this on the nutrition label, okay? Because this is how we talk about charge within these solutions, so that as we're hydrating the body, as we're putting these things back into our body and back into our blood, we're just understanding that all of these ions and electrolytes have a certain amount of charge, and we're focusing on the charge. Okay, what is a mole of charge? What is an equivalent? Okay, well, an equivalent is 1 mole of charge, and we know ions have charge. Okay, so if I were to ask you 1 mole of sodium cations, how many equivalents is this? Okay, all you're going to do is take the number from the mole, multiply it by the number on the charge. Obviously, there's an understood 1 here, and that's how many equivalents you have. So if I have 1 mole of plus 1 charge, then I have 1 equivalent. Okay, so all I did is I did the 1 times 1, and I said, okay, I've got 1 equivalent. That is 1 mole of plus 1 charge. Now, it could also be negative charge. It doesn't have to just be positive charge, okay? And we'll get to that on a practice problem in a minute. What if I have 1 mole of calcium ions, which each calcium ion has a plus 2 charge? So I'll take the 1, and I'll say times positive 2, which means I have 2 moles of charge, right? So I've got 2 equivalents. I'm taking the number of moles and I'm multiplying it by the number in my charge on my ion to get the number of equivalents. Okay, what if I have half a mole of calcium ions? Well, then I'll take 0.5 moles. I'll say times 2, which means I have 1 mole of charge. So I have 1 equivalent. All right. Hopefully that's not some terrible math. Hopefully that's pretty easy. But it is sometimes an uncomfortable idea for students to talk about a mole of charge. But I want to summarize right here. You may notice 1 mole of sodium ions has the same quantity of charge as half a mole of calcium. Why? Because the charge is double on calcium. So you only need half as much of it to give you the same number of equivalents. All right. There's also such a thing as a milliequivalent, which is abbreviated as MEQ. And these are used frequently because low concentrations of ions in the blood. Okay, so that's what this table shows you down here. Some of them, like it probably doesn't surprise you that sodium and chloride are present in the blood in larger amounts. We need sodium and chloride for lots of things. But then a lot of these other ions are present in lesser amounts. And these are all measured in milliequivalents per liter. Okay. I'm not going to ask you to memorize any of these numbers off this table. I'm just using it as an example to show you some things. Okay. All right, I want to work another problem or two on equivalence, and then we're going to get to our very last topic, which is osmosis. We're going to have to skip a few slides to get there. Okay, how many equivalents of aluminum cations are obtained from 1 mole of plus 3 cations? So again, you're going to say I've got 1 mole. And I'm going to say times, I've got a positive 3, so I've got 3 equivalents. Okay. Now, if I ask you, all right, I only need 1 equivalent. That's why I'm changing the question here. If I said I only need 1 equivalent, how many moles of aluminum plus 3 cations would give me 1 equivalent? You say, okay, well, I know 1 mole gives me 3 equivalents, so I just want 1/3 of that, right? So I would just take 1 mole and divide by 3, and I'd say, okay, 0.33 moles of aluminum plus 3 cations would give me one equivalent. Does that make sense? I hope so. Again, if you're like, no, that doesn't make sense, write that question down, email it to me, come see me, catch me after class on Thursday. Please, I want to know your questions from this lecture. Okay, now I told you it doesn't have to just be positive charge. So far, we've just been talking about all these cations. Well, we can also talk about anions because anions have a charge. And it doesn't say an equivalent is 1 mole of positive charge. It just says an equivalent is 1 mole of charge. So that charge could also be negative. So how many equivalents of fluorine anions are obtained from 1 mole of fluorine? Okay, so you do 1 mole, again, times the charge, times -1, and that is 1 equivalent. That is 1 mole of charge. Where it can get complicated is if I say, okay, what if I have aluminum fluoride, AlF3? How many equivalents do you have? If I have 1 mole of aluminum fluoride, how many equivalents is that? Well, you've already looked at 1 mole of aluminum cations is 3 equivalents, and 1 mole of fluoride anions is 1 equivalent, but we don't have the exact same numbers. If I have 1 mole of this substance now, I have, when that dissociates, I have 1 mole of aluminum plus 3 cations. How many moles of fluoride anions am I going to have? For every 1 aluminum cation, I need 3 fluoride anions. So if I have a whole mole of aluminum cations, I need 3 whole moles of fluoride anions. Okay? Now from here, I think you can get it. You would do 1 times the charge. That's a weird 3. Okay? And that would be 3 equivalents just from your cations. And then you do 3 times the -1, which is going to be not -3 equivalents. Really, you're just looking at the number here, which would be 3 more equivalents. So total, I would have 6, 3, and another 3 for my anions, 6 equivalents. Okay. Again, we're not saying these are negative equivalents. That doesn't make sense. An equivalent is just a mole of charge. Okay, and a negative charge is still charge. So 6 equivalents total. Okay. So if I give you what am I trying to say? A compound. You just break it up into its cations, and then you calculate the equivalents from that. All right, we are going to skip over properties of colligative properties, elevated boiling point, freezing point depressions, and we're going to go straight to osmosis. This is our last topic, and then we'll be done. I'm probably going over time, I bet. All right, osmosis. So I bet when you were in high school, this is what everybody joked osmosis was, right? Some of you probably tried it. It would actually be really awesome if this is how information was disseminated. If we could just sleep on our textbooks and learn. Get the information into our brains that way. But that's not how it works. So, osmosis is the passage of, let's define it, solvent, okay, through a semi-permeable membrane separating 2 solutions of different concentrations. That's a lot of big words. What does this mean? So first off, you know what solvent is, right? Because a lot of people get this part confused. They think it's the passage of the solute particles moving. It's kind of weird to think about the particles staying still and the solvent moving. Okay, but that's what osmosis is. It's the passage of solvent through a semi-permeable membrane. What is semi-permeable? Okay, look down here. If you have this picture, this black line right here would be it's dividing 2 sides of my solution, the solution on the left from the solution on the right. And a membrane, think about like a cell. Every cell has a membrane, right? And some things can pass through it and some things can't. And that's what it means when it says semi-permeable. Usually small things can pass through just fine with no help. A lot of the bigger things like proteins or things passing into and out of a cell have to have help, all right? So the things that don't need any help and that can pass right on through are permeable. And so when the membrane is selectively permeable, we say it's a semi-permeable membrane. All right, separating 2 solutions of different concentration, okay? And the last thing I want to say before I define and fill in these other blanks is that the solvent, make a note of this, always moves from low concentration well, you know what? I'm gonna make this a little more specific. The solvent moves from an area of low solute concentration. I'm gonna abbreviate the word concentration to an area of high solute concentration. Okay? It always moves from an area of low solute concentration towards or to an area of high solute concentration. That's the way the solvent moves. This right here is what we call a gradient. Okay? When you have a separation of concentration, you have a gradient. And you probably have even heard that in some of your biology classes before. So if you're looking at these pictures down here, this would be my area of low solute concentration. This would obviously be the area of high. The red dots, represent your solute. Okay, so we got low solute concentration on the right, high solute concentration on the left. All right. Now I want to come back up here to these blanks. Osmosis depends on the concentration of particles. We don't care about what the particle is. We just care our particle identity. We just care about how many particles there are. Okay? We think of all particles the same. So osmolarity, how do we define it? If you were to calculate osmolarity, because you can calculate osmolarity, it is the moles of particles divided by liters of solution. Okay, very much like just molarity, right, which is moles of solute per liter solution. But osmolarity is in particular moles of particles. Now, how the heck do we define a particle? What is a particle? It's actually really simple. Look at a substance like sodium chloride. If you have a substance that will dissociate, you're just going to count the number of particles it would dissociate into. This would dissociate into one sodium cation, and it will dissociate into one chloride anion. That is two particles. One particle, two particles. Okay, so if you have 1 molar sodium chloride, you just multiply the number of particles times your molarity to get your osmolarity. I'll show you that again, but you always figure out how many particles am I going to get from my compound, and if it helps, write those out, and then you count those number of particles and you multiply that by your molarity to get your osmolarity. Okay, what about glucose? If I have 1 molar glucose, we've already talked about the fact that glucose will not dissociate. It won't break down any further. It won't break apart into ions. So glucose is your particle. So that's why we're saying here 1 molar times 1 particle is just 1 osmol. All right. So some compounds like glucose, insulin, fructose, sucrose, these non-electrolytes, they're not going to give you particles, any more particles. They themselves are the particles, so their molarity is equal to their osmolarity. All right, so I want you to look at, again, these two solutions I have that are separated by this semi-permeable membrane. And on the right side, it says the concentration of particles is 1 osmol. On the left side, it says the concentration of particles is 3 osmol. I want to define 2 quick words here. Whenever you have a lower osmolarity, a solution with a lower osmolarity, we call this hypotonic. Hypotonic. Whenever you have a solution with a higher osmolarity, we call it, you probably can guess, hypertonic. Okay? Okay, so now if I ask you, if osmosis is going to run across this membrane and the solvent's going to move, which way would it move? And what would these pictures look like in the end? Which way would it move? Would the solvent move this way, or would the solvent move this way? To the right or to the left? So write your answer down. Some of you cheated and scrolled to the next slide. The correct answer is that the solvent would actually move towards the left, okay, to dilute the solution with higher osmolarity until you get 2 solutions with equal osmolarity. That's the way osmosis works across the gradient. So now we have 2 solutions on either side of our membrane with equal osmolarity, and when you have equal osmolarity, we say the solutions are isotonic. That's the blank up here at the top.
same osmolarity, same number of particles per liter. And I don't have a poll question. Okay. All right. If you have questions on this, like I still don't understand how this works, or why did the solvent move from right to left or whatever, write those down, please, and email them to me. Or what I actually prefer is that you come see me with them so I can discuss them with you in person and not via email. I hate discussing stuff in email. Sometimes I do it, but I prefer in person. All right, let's practice calculating osmosis, and we're almost done. Okay, when calculating osmolarity, ask yourself, how many particles or ions will this compound produce? And then you're going to multiply that by your molarity, okay? So let's say we have these compounds here, 0.5 molar of potassium chloride. This is going to break apart into potassium and chloride, which will be a negative. So I will get 2 particles, right? So I'm going to multiply this times my molarity and say, okay, I'll have 1 osmol. Or it would be 1 osmolarity. What about the next one? All I'm doing is I have 1 molar calcium chloride. Okay, calcium chloride is going to break apart into 1 calcium, right? I'm not looking at the charge. With equivalence, you would focus on that charge, right? But here, we don't care about the charge. We just care about how many particles. So I'd get 1 calcium and how many chlorides? I would get 2 chlorides. Total, 1 plus 2 is a total of 3 particles. We multiply the number of particles times our molarity, and that is 3 osmoles. Okay, can you do the last one ahead of me? Maybe pause the video and tell me what the osmolarity of 3 molar glucose solution would be. This is just going to give me 1 particle, right? Glucose is the particle. So obviously then times your molarity is just 3 osmoles. Because glucose is one of those biological molecules that does not break apart or dissociate into ions. Okay. So that is some example and some practice with calculating osmolarity from compounds. There's some more really good practice problems in the back of chapter 9 in the textbook. Okay, osmosis. So we have these pictures of some red blood cells here. Last 2 things on osmosis, this one, and then we're going to talk about one more slide. And it says we have red blood cells in isotonic solution, and then look, red blood cells in hypotonic solution and red blood cells in hypertonic solution. And obviously the blood cells look different in each solution. In the isotonic solution, they look like a normal red blood cell. They kind of look like a cushy, kind of like a donut, right? They have kind of like this dip in the middle here, which is the dark shaded region. And that's how red blood cells should look. But they look different in the hypotonic solution. They look swollen, okay, like kind of way more puffy than the ones on the left here. And why is that? I always have to change my color of my pen because red won't work on this picture. But I'm going to draw on this picture, and you should too. So for a hypotonic solution, what does that mean? That means the solution outside of these cells has a low solute concentration. And that means inside the cells we have high solute concentration, okay? Now, we have a gradient because we have a separation between our concentrations. They're not the same. So which way is the solvent? We have solvent in the blood cell and we have solvent outside the cell. And the extracellular fluid is what we call that. Which way is it going to move? Remember, it always moves from an area of low solute concentration towards an area of high solute concentration to try to dilute it. So simply doing its job via osmosis. Oh, red does not work here. I think white actually works the best. It's going to rush in. All this extracellular fluid is going to rush into the cell, which is what causes the cells to swell. And if it rushes enough in, it would cause the cell to burst. Okay. And then why do the red blood cells look like this in hypertonic solution. These are shriveled. And then I'm going to write that they're swollen over here. So we had swollen red blood cells in hypotonic solution. Why are they shriveled in hypertonic solution? Well, how is the concentration difference working? If they're in hypertonic solution, what that means is the solution outside the cell has high solute concentration. And that means the area inside the cell has low solute concentration. So the solution is going to rush from low to high. So all of the solution is going to rush out of the cell trying to dilute the extracellular fluid. And when it does that, it leaves the red blood cell all shriveled like this, which is not good either, okay? So that's why it's important that our blood and our cells maintain proper equivalent levels, proper amounts of charge, proper amounts of solute in isotonic solution. Okay, again, any questions, write them down, and please come to me with your questions. Last slide here, and dialysis is just an example of what we're going to talk about, osmosis and oxygen transport, okay? So I've got two pictures. I've got your arterial veins and then your venous veins. Obviously, follow the white arrows. Our arterial end, these are veins coming from, they're freshly oxygenated blood, okay? Coming from the heart and lungs and being pumped throughout the rest of the body. So blood that is coming through these veins has a high concentration of oxygen. So I'm going to put an upward arrow, meaning a high concentration of oxygen. And it has just picked up oxygen from the lungs, and now it's going to be pumped by the heart through the rest of the body. Okay, so high concentration of oxygen, and it has a very low concentration of CO2 because we know at the lungs we exchange, CO2 is removed and exhaled, and then all the oxygen we're breathing in goes into our red blood cells, into our blood. Okay, so this is the conditions of our freshly oxygenated blood, and then that's going to be pumped throughout the body to our tiny blood vessels, which are known as capillaries. And this is where the exchange of oxygen and CO2 happens with our surrounding tissue and cells, okay? So I want you to notice a few things. Inside this capillary, you might notice these like purple circles. These are representing like red blood cells, okay? And then you might notice all these little dots right here, these little dots, which is, this capillary is a semi-permeable membrane. Okay, so through all these dots is where the exchange of things like oxygen and CO2 happens, okay? So we also have proteins. I want to acknowledge this. We have proteins down here in our blood, but proteins are really, really big, okay? And they cannot pass through these little teeny dots the way small molecules like oxygen and CO2 can. You can almost think of oxygen and CO2 as the solvent here. It's kind of weird to say. Not that they are the solvent, but they're small enough that they can pass through these capillary holes in our capillary like solvent can, okay? Now, larger things like the red blood cells themselves and the proteins cannot. They stay in the capillary, and then they move on through the venous veins and things back to our lungs, all right? But the oxygen and the small molecules like CO2 can travel in and out. They can passively move in and out. So their movement is going to be directed by a gradient. Okay, and you might notice, let's write this out, in our surrounding cells and tissues, these cells have been hard at work, and so they have used up any oxygen that was given to them, and they've created a lot of waste. So in the cells, we have a situation that looks like this: low oxygen content and high amounts of CO2. Whoops. Because they've been doing things, making things. That's what our cells do. They're machines that are constantly making proteins or replicating DNA, things like that. And in doing all these things, we make CO2 and we use oxygen. Okay, so these are the conditions in our cells. These cells need oxygen and they need to get rid of CO2 waste. Okay, now in the blood, what we said is we have high oxygen, low CO2. So you can see that oxygen is going to move from an area of high concentration in the blood is going to be forced and pushed out to an area of low concentration down here in these cells. CO2 is a high concentration in the cells, and it's going to be moved. Oh, I'm on eraser. It's going to be moved, follow these arrows into the blood where there's a low concentration of CO2. And that's the exchange we want to happen, right? We want to continuously deliver oxygen to the cells because they need it, low concentration of oxygen in the cells. So we're going to deliver from the blood to the cells all of our oxygen and other things, nutrients, other raw materials. And the CO2 is going to be pulled via gradient from the cells back into the capillaries, and then it's going to be delivered out to the lungs and exhaled. Okay? So that is how oxygen transport works. It's more detailed than that. It involves hemoglobin and things like that, but unless you're in a biochemistry class, we're not going to get into how hemoglobin works, which is really fascinating, but not a conversation for here. So last slide here. This is how dialysis works. People who have kidneys who can't filter out their blood for them, kidney failure, they will have their blood removed from their body via a dialysis pump. That pump will then take that blood, pump it into a dialysis solution. Okay, so in the blood, let me say here, in this blood we have high concentration of waste. And in the dialysis solution, which is the blue here, we have low, or I should say no waste. Okay, so that waste in our blood is going to be filtered out of the blood into the dialysis solution, and then we filter it back into the body. Okay. All right, that's it for solutions. That's all of Chapter 9. I have no idea how long this is, so if I went over time, I apologize. But we will recap on any of this at the beginning of class Thursday, and we will start Chapter 10 on Thursday as well.